So the big challenge of cryobiology, the freezing of living tissues and cells, is ice formation. The whole field is basically dedicated to finding ways of preventing ice formation. The reason that ice crystals are so bad for biological material is that they're sharp and spiky. And so when you freeze something and ice forms inside of it, it tends to break up the structure. The big deal about this paper that was just published by Mary Hagedorn and Boris Rubinsky's lab is that Boris's lab has been working on a new way to do cryobiology. It relies on a rigid container that you put the sample in to prevent ice from being able to expand into crystals. Isochloric freezing involves putting something in a container that doesn't expand, and so when the crystals try to form, they can't. And that allows you to freeze things that are three-dimensional. And so now we can start thinking about freezing tissues that are part of organs or structurally complex material. And so we went looking for a willing collaborator for Dr. Rubinsky, and Mary Hagedorn, of course, stepped up, realizing that the ability to freeze large fragments of coral instead of sperm or eggs or larvae would really make it much easier to bank all the species of coral that we're trying to rescue right now. The problem with coral sperm is that it is only released during a spawning event, which happens like one night a year. And so on that one night, you have to figure out what night that's going to be. I'm sure that's not super easy. And I had to get there, and you had to be there to collect the sperm cloud. The ability to freeze fragments, on the other hand, you could collect anytime. And so now they've demonstrated that we can, in fact, bank these coral fragments, which is a big deal because now Mary doesn't need to go out on the special night of the year and know which coral is going to release its sperm. She can go anytime, anywhere, and get all of the endangered species of coral into a biobank much more quickly.